Anomalocaridid, or Anomalocarididae, is a family of radiodonts, an extinct order of stem group arthropods most prominent during the Cambrian period. Historically, Anomalocarididae was used as a catch-all group for nearly all radiodonts, including forms now placed in several distinct families. Modern analyses have restricted Anomalocarididae to a much narrower clade. Now in this video, I'll focus on the stem branch leading to the Amplectobeluidae, Anomalocarididae, and Tamisiocarididae radiation, a lineage I'll refer to as Anomalocaridid-grade Radiodonts. Anomalocaridid-grade Radiodonts stand out from other primitive Cambrian creatures thanks to their anatomical sophistication, a pair of large raptorial frontal appendages that were modified to serve different feeding functions across the group. Streamlined bodies propelled by coordinated waves of lateral flaps enabling active, high maneuverability swimming, and advanced compound eyes rivaling those of modern arthropods. In this video, I'll present every Anomalocaridid-grade Radiodont ever described, including possible synonyms and dubious names, sorted based on their estimated size, from the smallest to the largest. Lenisicaris. Materials of Lenisicaris consists of only its frontal appendages, which are robust and notably lacking auxiliary spines, distinguishing it from many other radiodonts. The genus Lenisicaris was named in 2021, which includes the type species Lenisicaris lupata, found from the Maotianshan shales of China, and the reassignment of 3 specimens from the Kansas Formation of the US, originally described as Anomalocaris pennsylvanica, to Lenisicaris pennsylvanica. Body proportion comparisons with Anomalocaris suggested it was a small nectonic predator inhabiting shallow marine environments and preying on soft-bodied organisms. Paranomalocaris is a radiodont with problematic placement. The genus was described in 2013, based on fragmentary materials, consisting of only frontal appendages. They were notably elongated and bore multiple well-developed endites. The segmentation and spine arrangement imply a grasping and trapping function. 2 species are known, both from the Guanshan biota of Yunnan, China. Lyrarapax is a small radiodont, with the type species only reaching around 8 cm in length. Lyrarapax was described in 2014, based on multiple well-preserved specimens found in the Maotianshan shales in the Yunnan province of China. It possessed a pair of raptorial frontal appendages, a limbless trunk with lateral swimming flaps, dorsal gills, and terminal tail streamers. The frontal appendages were short, with a pair of large, claw-like first endite bearing multiple spines, suited for grasping prey. The first pair of trunk flaps were greatly enlarged and paddle-shaped, suggesting enhanced maneuverability, with the flaps decreasing in size posteriorly. The fossil of Lyrarapax was exceptionally preserved, with its nervous system imprinted on the stone, revealing that the frontal appendages were controlled by the protocerebrum, similar to the antennae of velvet worms. This discovery provided key evidence for understanding the evolution of the arthropod. Lyrarapax lived in a diverse, well-oxygenated shallow sea alongside early arthropods, lobopodians, priapulids, and other radiodonts. Innovatiocaris is one of the few radiodonts known from a nearly complete body fossil. The fossil was first discovered in the early 90s from the Maotianshan shales in southwestern China. The specimen was fully described and formally named as Innovatiocaris in 2022. Originally, it was referred to Anomalocaris. 2 additional taxa are known solely from complete frontal appendages. The nearly complete holotype fossil represents the juvenile individual measuring 15.2 centimeters. It provided a rare insight into the overall anatomy of early stem arthropods. It possessed 10 pairs of flexible main swimming flaps along its trunk, 6 pairs of smaller lobes on the neck, 3 pairs of large tail fans, and a pair of long tail streamers. Its eyes were large and stalked. Notably advanced eyes were a shared characteristic of radiodonts. Innovatiocaris was likely a nectonic predator, using its spined frontal appendages to seize soft-bodied or lightly armored prey in the water column. The Chengjiang paleo-environment represents a shallow marine setting with rapid burial and low oxygen conditions that promoted exceptional fossil preservation. Lyrarapax is a newly described genus of radiodont. 2 specimens are known, consisting of only frontal appendages, which were found in the Kansas Formation in Pennsylvania. Lyrarapax was named and described recently, in late 2025. Its frontal appendages exhibit a unique feature, which indicates that Lyrarapax may be an intermediate form between bottom-feeding radiodonts and the suspension-feeding radiodont Tamisiocarididae. Lyrarapax likely hunted by sweeping the loose substrate on the seafloor to catch soft-bodied invertebrates. Guanshancaris was first thought to be a species of Anomalocaris. Subsequent studies revealed that it differed significantly, leading to the naming of a new genus, Guanshancaris, in 2013. It is only known from fragmentary materials, primarily its frontal appendages and oral cone, which were discovered in the Guanshan biota in southern China. Guanshancaris exhibits robust raptorial frontal appendages and a comparatively more powerful oral cone, suggesting a diet consisting of hard-shelled prey such as early arthropods. It is likely that Guanshancaris was an active nectonic predator. Shucaris is a recently described radiodont. It was formally named in 2024 based on multiple fossil materials that include frontal appendages, a nearly complete body, and an unusually small oral cone. These specimens were found in the Mount Tianshan shales of Yunnan Province, China. Shucaris exhibits a classic radiodont body plan with carapace-like structures, one of the defining traits of Amplectobeluidae radiodonts. This structure, along with the robust frontal appendages, indicate a raptorial function, adapted for grasping and processing hard-shelled prey. Traditionally, Shucaris has been recovered as a member of the Amplectobeluidae, closely related to Ramscoldia and Amplectobelua, but workers who describe the taxon found it to be within Anomalocarididae. Caryocyntrips is a dubious radiodont. It was first named in 2010 from a fragmented fossil found in the Burgess Shale Formation of Canada. 3 species of Caryocyntrips have since been named, including one from a much larger specimen found in Spain. All known specimens of Caryocyntrips consisted of only its frontal appendages, which is thought to have been used in a scissor-like grasping or slicing motion. Most studies found that Caryocyntrips may sit outside of the monophyletic Radiodonta altogether, but in a recent paper describing Shucaris, workers placed it within Anomalocarididae, closely related to Lenisicaris and Paranomalocaris. Tamisiocarididae was the first radiodont discovered to be a filter feeder. It was discovered in the Sirius Passet Lagerstätte in North Greenland and was first named as Tamisiocarididae in 2010. Morphologically, Tamisiocarididae differs from classic raptorial radiodonts. Its frontal appendages were long and slender, composed of numerous podomeres bearing densely packed, elongated endites with fine auxiliary spines that form a comb-like structure. They create a filtration apparatus capable of trapping very small particles. This implies that Tamisiocarididae was clearly adapted for suspension feeding, and it demonstrates that complex trophic partitioning had already evolved among radiodonts early in the Cambrian. Although the body is incompletely known, Tamisiocarididae conformed to the radiodont body plan with lateral swimming flaps, dorsal gills, and a tail fan. Size estimates suggest a total body length of 40 to 50 centimeters, making it a relatively large radiodont. Hurdiidae is a problematic genus of radiodont known from deposits in China and the western United States. Hurdiidae has been variably assigned to different radiodont families, and its monophyly remains debated. The genus was erected in 2021 to accommodate the species previously assigned to Anomalocaris. The type species, Hurdiidae saron, is known from the Maotianshan Shale or Chengjiang biota of Yunnan, China, and is represented only by frontal appendages. A specimen once thought to be a complete body was later found to belong to a different radiodont, now assigned to the genus Innovatiocaris. 2 additional species have been tentatively referred to the genus, Hurdiidae magnabasis from Nevada and Hurdiidae consimilis from South China and possibly California. Ecologically, Hurdiidae was likely a nectonic predator, using its robust frontal appendages to grasp prey in the water column. Their occurrence in the Chengjiang type and Laurentian shallow marine environments suggests they inhabited warm, productive Cambrian seas, contributing to the early diversification of radiodont feeding strategies. Anomalocaris was a large-sized Tamisiocarididae radiodont discovered in Australia. It is primarily known from its frontal appendages, which dated to the Middle Cambrian. Anomalocaris was originally described as a species of Anomalocaris before a more detailed analysis found it to be a distinct new genus in 2023, classified within the family Tamisiocarididae. Anomalocaris' frontal appendages bore long, slender spines, lined with numerous fine auxiliary bristles, an arrangement well suited for trapping small organisms in the water column. This indicates that Anomalocaris was a nectonic suspension feeder, filtering small prey in the water while steadily swimming. Its mouth was a triradial oral cone, consistent with other Tamisiocaridids. Fossil eyes attributed to Anomalocaris were large, non-stalked, and composed of 13,000 unusually large lenses, a design likely adapted for low-light conditions. Anomalocaris is the first described radiodont and one of the largest Anomalocaridids, reaching roughly 50 cm in length with possible larger species. Its size and anatomy suggest it as one of the earliest known apex predators. Anomalocaris was described in 1892. The name was not given to the radiodont we know today. At that time, it was only known from isolated frontal appendages, which were mistakenly identified as the abdomens of shrimp-like crustaceans, hence the name's meaning, abnormal shrimp. Other body parts were later described separately and misidentified as unrelated animals, including a jellyfish-like fossil later named Peitoia. It was not until the late 20th century that these species were recognized as parts of a distinct type of animals, which then also revealed that Anomalocaris, Peitoia, and other related forms belong to a same group, now known as Radiodonta. Today, 2 species are confidently assigned to Anomalocaris, the type species A. canadensis and the possibly larger A. dahliae. Anomalocaris swam by undulating flexible lateral flaps along its body. 13 pairs of main flaps on the trunk and 3 pairs of smaller lobes on the neck. The tail was a large fan, composed of probably 3 pairs of large lateral fin-shaped lobes and 1 terminal lobe-like tailpiece. This structure, combined with its advanced dragonfly-like compound eyes and its armored, spiny frontal appendages, indicated as a capable, active, free-swimming predator. It had a unique disc-like mouth, known as an oral cone, composed of several plates organized triradially. These oral cones are distinct from each other among the members of Radiodonta. Recent studies suggest that it primarily fed on small, soft-bodied animals. Anomalocaris had an almost global distribution. Fossils have been found in Canada, the US, China, and Australia. It inhabited warm, tropical seas, from the shallow reefs to the deeper offshore environment in the early to middle Cambrian period. Lenisicaris was a large radiodont with an uncertain phylogeny. It is known from isolated frontal appendages found in the Chengjiang biota of Yunnan province, China, with similar materials also reported from the Kansas Formation of Pennsylvania, United States. Lenisicaris was first described in 2018, based on fossils recovered from fine-grained mudstones of the Cheongchusu Formation. Morphologically, Lenisicaris is defined by its exceptionally large frontal appendages, the largest measuring around 28 centimeters long, implying a total body length of approximately 75 to 100 centimeters. The size and structure of the appendages indicate an active feeding strategy, possibly targeting relatively large and mobile organisms. Lenisicaris has a highly debated position within Radiodonta due to its fragmentary nature and combination of ancestral and derived traits. Anomalocaridid was the second genus of radiodont found to possess natobase-like structures, or GLS, and an atypical irregular oral cone, indicating a more complex feeding apparatus than that of most radiodonts. Anomalocaridid was first described in 2018 from the Zhenjiang biota in China. It is only known from fragmentary remains. Its frontal appendages had prominent auxiliary spines, suggesting a grasping function. Two species of Anomalocaridid have been described, the holotype R. platycantha and the taxonomically problematic R. consimilis, the holotype being much larger among the largest known Amplectobeluidae radiodonts, reaching 130 centimeters long. Anomalocaridid was likely an active nectonic predator, feeding on soft-bodied or lightly armored animals in shallow, well-oxygenated marine environments. Fossils of Anomalocaridid occur alongside other radiodonts, such as Amplectobeluidae and Lyrarapax, indicating it was part of a diverse predatory guild in the Cambrian seas. Amplectobeluidae was a giant Amplectobeluidae radiodont. The type species reached almost 1.5 meters long, exceptionally large for Cambrian standards. It was described from fossils discovered in Yunnan Province, China, with a second, much smaller species described from specimens found in Burgess Shale, Canada. It was initially described as a species of Anomalocaris in 1992, but was redesignated as a new genus named Amplectobeluidae after more materials became available. Amplectobeluidae had highly specialized frontal appendages, which functioned like pincers. They basically invented opposable thumbs 500 million years early before it became popular. The forward-hooking spine enabled strong grasping to catch prey. Amplectobeluidae was the first radiodont found to possess natobase-like structures, or GLS, beneath the neck, which could rotate and shred food, similar to the chewing structures of later arthropods. Its mouth also differed from typical radiodonts, lacking a peitoia-type oral cone and instead bearing multiple flat tooth plates. These traits are the defining features of the Amplectobeluidae family. The body bore 11 pairs of elongated flaps that decreased in size towards the rear, with smaller flaps on the neck and long tail streamers at the end. Amplectobeluidae was an nectonic apex predator, using its powerful appendages to seize prey or tear pieces from larger carcasses. It served a dominant predator role in the Cambrian ecosystem. Those are all the Anomalocaridid-grade Radiodonts that have been formally described to date. They represent the first truly complex marine predators in Earth's history, with most of them occupying the role of apex and mesopredators, preying on a wide range of soft-bodied nectonic animals, and possibly exerting strong selective pressures on early arthropod defenses. The Anomalocaridid-grade Radiodonts changed the way we view early Cambrian ecosystems, with their remarkable functional and ecological dominance during the Cambrian explosion, exhibiting some of the most sophisticated sensory and predatory capabilities far earlier than once thought possible. They evolved into some of the largest animals of their time, dramatically outsizing their invertebrate contemporaries. Their size, speed, visual acuity, and advanced grasping limbs allowed the Anomalocaridid-grade Radiodonts to exploit a wide range of trophic strategies and reign supreme in the planet's newly born, primitive marine ecosystems.